All right, so now I think we have the next talk, our first group talk coming up. It's going to be the in vitro human cell types group talking about the lineage. Boas coming up here, Sherman Koo, Vilas Menon, Refugio Martinez, Zizhen Yao, John Mitch, and yeah, Boas. You have the floor.
Yeah, we'll be at the podium. Oh, okay. I think we'll sit here. Hello, I'm Boaz Levy, and it's my pleasure to introduce the work from the in vitro human cell types group, a human brain lineage tree from embryonic stem cell derived neurons. And the project that I'm going to present started off with two major goals. First was to build a genetically tractable system to understand the mechanisms of human cortical development, lineage, and maturation. And secondly, to help establish a robust and well-validated platform for the IPS and the human embryonic stem cell field to understand the basic cell biology of human brain development. And we are interested in understanding human brain development because there are significant differences in the developmental pathway between mouse and human. In human, as well as other primates, there's a greatly protracted period of neurogenesis. There are regions such as the outer subventricular zone, which are transient and appear in human and are not present in mouse. And in these regions, there's also cell types that exist, which are poorly modeled in mouse. So the goal of our group was to take undifferentiated human embryonic stem cells and to coax them through this process to create cortical neurons. So in doing so, we faced several major challenges. We had to be able to engineer stem cells to report on their molecular identity. We had to develop methods to generate cortical cell types. We had to measure cells at single cell resolution. We had to be able to compare our cells that we were generating with primary tissue in order to understand what we're really creating. We had to be able to take this gene expression data and infer a lineage tree from this data. And finally, we wanted to be able to experimentally test what we had inferred. So our team talk today will be composed of several portions. Ref Martinez will talk about how we're making these cells through engineering reporter lines as well as bringing in-house differentiation protocols. Zizhen Yao will be discussing how we measure these single cells. John Mish will be discussing how we actually take this gene expression data and actually compare it with previous atlas data and primary tissue to understand what we're creating in a dish. Vilas Menon will describe how we take the single cell gene expression data and infer a lineage tree based on this. And Sherman Koo will describe how we actually experimentally test that predicted lineage. So without further ado, I'd like to hand it over to Ref Martinez, who will discuss how we're making cells.
Thanks, Boaz, and good morning. So as mentioned, one of the goals of our group was to develop an in vitro protocol, starting with human embryonic stem cells, to mimic cortical development, specifically using morphogens that are known to influence the derivation of excitatory or inhibitory neurons. And to that end, we established a protocol that spans 54 days and highlighted key time points shown here in this gray area that will be referenced throughout our talk. In the beginning, we start with the human embryonic stem cells at day 0, and we transition them through 3 major phases, the first being the telencephalon induction phase, followed in the second phase by the expansion of the neuroprogenitor cells. And in the final phase, these cells are further differentiated and matured into either excitatory or inhibitory neurons. Now, this results in a very heterogeneous mixture that includes both cortical progenitors and neurons. So over the course of our differentiation, we use ICC to kind of track the emergence of these distinct cell types chronologically. So early on, we can see the expression of FOXG1, which is a marker of rostral progenitor cells. Later on, we can see the expression of the intermediate progenitor cells as marked by TBR2. And at the later stages, we can see the expression of layer-specific markers, as in the case of TBR1, which is a lower layer marker, and the upper layer marker CUX2, and the expression of somatostatin specifically for interneurons. Looking at gene expression data pretty much confirms that our methods are highly reproducible, as populations of cells are represented here in individual colors, tend to segregate and cluster together based on the time points at which they're harvested, and separate from those other populations that were harvested at other different time points. We use electrophysiology to test that the neurons that we are making are in fact functionally active. And so we recorded and confirmed that some of our cells are in fact showing spontaneous activity and firing patterns that are typical of neurons that have matured enough to form the proper synapses and network connectivity. So convinced that our protocol was working well, we then switched the focus to developing these reporter lines. And we've made several of these reporter lines that target genes that are important in cortical development and result in these fusion proteins where we have a fluorescent marker that is fused to the protein of interest. And these are the two lines that are most readily used and extensively used in our group. SOX2 is a marker for progenitor cells, and double cortin marks immature neurons. And so the true value of these reporter lines is in the ability for us to then track the emergence of these rare cell populations via fluorescence and to isolate those cells and further enrich for those populations. For example, in the case of double cortin, we can see that the double cortin fluorescence expression, the cells tend to increase in numbers over the course of the differentiation, and we can successfully kind of isolate and with FACS sort out these cells even when they are present only in low numbers, such as in day 12 and day 19. So with that, I'll end by summarizing that our group has successfully developed a very robust method to generate functional cortical neurons, starting with human embryonic stem cells, and that we can also make several reporter lines to help us enrich for those rare cell populations that we can then profile. And so next, Zishan will talk more about how we can further profile those cells using single-cell transcriptomics.
Thank you, Red. So to keep track of the whole development process of cortical development, we isolate in vitro cells at different stages of development, starting from day 0, the embryonic stem cells, all the way to day 54. So during this time, the in vitro cells progress from cortical progenitors to intermediate progenitors and immature neurons. As Reb mentioned previously, the neuronal cells are very well at the beginning stage of development. So we use DCX lines and the SOX2 lines to sort out this rare population to make sure that we have sufficient sampling of both progenitors and neurons. Okay, so this is that. So over the past 4 years, our group has established a very developed from scratch a very robust and highly efficient pipeline for single-cell transcriptome profiling. To assure quality, the in vitro cells are first tested by ICC and flow cytometry to make sure that we have presence of the key neuronal markers. And then from these cultures that pass to QC, we dissociated single cells, and then the cells are sorted by the fax machine into corresponding neuronal or progenitor populations. These single cells are then sequenced, and the resulting single-cell transcriptomic data are then sent to the downstream analytical pipeline for cell type characterization. So let's take a quick look at the data. And so in the global scale, the the principal component analysis indicates that our single cells are first separated into progenitors and neurons on the first principal component, and then they're separated according to the temporal progression on the second principal component. So to categorize cell types at a given time point of a given population, we use WGC algorithm to identify co-expressed gene modules as features to cluster single cells. Interesting, many of the cell type markers identified are important transcription factors and known to be involved in the patterning and cell fate determination for brain development. So, for example, let's zoom into the day 54 D6 positive cells. We have largely found 5 major cell types, and these include the interneuron cluster marked by DLX cluster. DLX gene family. And for the remaining excitatory cells, they are largely split into the ALX2 positive cells and the brain2 positive cells over here. Okay? So the anti-correlation of these gene modules are very prominent for this set of cells. And for the remaining, the post-3F2 positive cells, they can further cluster into the Hox positive cells and the ROBO3 positive cells. So we'll apply the same algorithm to all the populations at all different temporal stages, and in total we identify 43 different cell types. So to validate the cell types we identified based on the transcriptomic data, we validated some of the key cell type markers using the ICC. And this figure shows the staining of the post-3F2 and ALX2 cell type clusters and genes. And here we can see a very clear mutually exclusive gene expression of these two markers, which is very consistent with our single cell data. And they have very interesting spatial organizations. So to understand what these in vitro cell types mean and what they correspond to in the in vitro system, we have done comprehensive comparison of the in vitro cell types with the primary brain tissues. And John will tell you next what we have learned.
What is the identity of the cells that we've generated in culture and been able to profile? And furthermore, do they correspond to any known physiological cell types? So we first started off to address this question by taking advantage of the human prenatal laser microdissected atlas that was generated here at the Institute and given to the community. This is a rich resource that has transcriptomic information of multiple regions of the human brain that were dissected and analyzed. We focused our analysis on the earliest stages that were available, 15 to 16 weeks post-conception, and we just asked a simple question: which of the cell types that we have and which of the brain regions that we have information on have the highest correlation? So here on this plot, I'm showing you a heat map of correlation coefficients between our day 54 neurons at the terminal stages of differentiation along this axis, according to Zigen's classification scheme. And on this axis, parts of the human brain, prenatal human brain, organized from rostral parts of the brain like cortex to caudal parts of the brain like midbrain and hindbrain. Regions of blue indicate weak correlation, whereas regions of red indicate strong correlation on a selected sample subset of genes that are differential across the cells and regions. And what we can see from this unbiased analysis is a striking result. The LHX2 types of cells that we've generated in the warmer colors have high correlations to rostral parts of the brain like cortex, but weak correlations to caudal parts of the brain like midbrain and hindbrain. In contrast, the POW3F2 types of neurons in the cooler colors have weak correlation to the rostral parts of the brain like cortex, but strong correlations to parts of the caudal brain like midbrain and hindbrain. So this suggested that our LHX2 neuron types that we've generated are cortical neurons, whereas the POW3F2 types of neurons that we've generated are caudal types of neurons in the brain. To test this hypothesis directly, fortunately we were able to obtain actual human mid-gestation tissues from cortex and hindbrain that we were then able to inspect for the presence of our cell types. So in these diagrams, I'm showing you a schematic representation of the different stereotyped organization in these two tissues in the human brain. And on these diagrams, I'll show you the presence of our cell types that we've been able to discover. We first started looking at markers of the LHX2 types of cells, CTIP2 and NFIA. And what we saw was a striking difference between those two regions, where we saw many abundant CTIP2 expressing neurons throughout the cortex and multiple abundant NFIA expressing neurons throughout the cortex, but only very few and very weakly expressing neurons in the hindbrain marked by these markers. This suggests that the LHX2 expressing neuron types really are cortical neuron types. In contrast, we also looked at markers of the POW3F2 groups of neurons, such as TFAP2B and ROBO3, and we saw the opposite result, where there were abundant neurons throughout the hindbrain, marked in different patterns, but few, if any, were detected in cortex, suggesting that the POW3F2 neurons really are caudal brain neuron types. Now, the results that I've shown you just now demonstrate, number one, that the cells that we've generated are really physiologically relevant, and also the differences among them reflect differences in regional patterning in the brain. In the next segment, Vilas is going to tell you how we've assembled our single-cell RNA-seq data on all these cell types into a single unified lineage tree of human brain ontogeny.
So as John mentioned, we now have these cell types, or putative transcriptomic cell types, that we've identified from the single-cell RNA-seq data. And the next step is to try and see if we can actually infer a lineage relationship among all these different cell types. So can we actually identify which cell types are putatively arising from which progenitor types? So there are several challenges in order to do this, and the biggest one is, of course, that given a set of clusters, we now have to determine a distance among them. And identifying a distance metric when you have discrete cell types is actually not a very trivial operation. So using all the genes, or even a large subset of genes, like using a method like PCA or WGCNA, will identify clusters of cells for you, but not necessarily a good distance metric to actually identify the relationships among those clusters. So in order to address this problem, we've actually used a relatively conceptually simple but elegant method developed by Sharad Ramanathan in his lab. And the general idea is this. If you have 3 different cell types that you've already identified, and then you look at gene expression, and you find that there's a specific gene that's expressed in 2 of them, but not in the 3rd type, gene A, and then another gene, gene B, that's expressed in these 2 types, but not the 3rd type. What this tells you is that given these 3 cell types, cell type 2 and 3 are actually likely to be further away from each other than they are to this intermediate cell type 1. So what we actually do now is take all triplets of our cell types, look at individual genes, we're looking specifically at transcription factors to start off with, and then identify for each triplet, is there a relationship among the 3 types that we're looking at? So we've developed a Bayesian formulation of this idea. Actually, it's been developed by Leon Furstgott, a grad student in Sharad's lab. We've been using a Bayesian formulation of this idea to get a confidence measure for every triplet. And once we've gotten these triplets and their connections, what we can do is assemble the entire set of cell types into a tree that we have as our first putative excitatory tree from this protocol. Once we've done this, we can then layer on information as to what time point did these different cells come from. Does that make sense in terms of the progression of the lineage as we see it in the tree? We can also layer on information about whether these types are progenitors or postmitotic cells, and then again, look to verify whether things make sense. Do we see progenitors giving rise to different types of postmitotic and neuronal cells? We can also look back at our gene markers for our various clusters, and we find that the major split in our tree, unsurprisingly, happens at somewhere between day 19 and day 26, where we get these 2 broad classes of cells that Zajen and John talked about. And finally, most interestingly, for each of the branch points that we see in our tree, since they come from an individual triplet, we also get a list of candidate transcription factors that could be responsible for some of the fate decisions that are governing each of these splits. However, at this point, everything that I've talked to you about building the tree has been in silico, so this is all a prediction. So anything that we can that comes out of this algorithm has to actually be tested. And so in the next section, Sherman's going to talk a little bit about some of the methods that we're working on to test this putative lineage tree.
Thank you, Vilas. So we've taken 2 complementary strategies to validate our predictions. One is a clonal differentiation approach that's been spearheaded by John Mitch, and a second strategy that I'll talk about later is barcoded viral lineage tracing. So the main goal of clonal differentiation is to assess the differentiation potential of our day 26 cells and to see what they can become. And to do this, what we can do is to sort out single cells from our day 26 cultures, separate them either based on prospective markers or don't separate them at all and just do so blindly, allow them to differentiate over time, and at day 54 we can profile them by immunostaining for various key markers, and in that way we can correlate these progeny to the predicted cell types. And the main point I want to reiterate here is that this strategy tells us the potential of the progenitors. So based on John's experiments, we do in fact see that single cell colonies can generate distinct cortical cell types. So shown here are 2 single cell colonies that are immunostained for various markers. In blue are human nuclei and neurons, and in green and red are these key markers that allow us to uniquely identify or distinguish between our predicted cell types. As Vilas mentioned, we know that LHX2 marks this branch of our predicted tree, and POW3F2 marks that branch. And so if we quantitate, for example, CTIP2 over thousands of single-cell clones, we do see that CTIP2 is highly correlated with LHX2 positive colonies and not correlated with LHX2 negative colonies. And all this is, in fact, consistent with what our predictions are. Now, the second strategy, barcoded viral tracing, is based on a technique that's been used for a long time, in which we can label a cell of interest and watch it over time and see what types of cells it gives rise to. And indeed, in our cultures, what we see, at least at a first approximation, is that we have two distinct clone types. One type contains cells that are distinctly non-neuronal, and another type of clone contains cells that have very distinctive neuronal-like morphology. Now, looking at morphology alone can be limiting. So to get around this, what we've done is to design a retrovirus that expresses TD tomato along with a degenerate 10 base pair barcode located on the same transcript, such that it's detectable by RNA-seq. So what this allows us to do is to infect our cells of interest, allow them to differentiate, collect them, sort out the infected cells, analyze those cells by RNA-seq, and in that same step, we can read off the 10 base pair barcode, then apply our computational methods, and in the end, we can map those progeny to our various predicted cell types. And based on looking at the barcode, we can then say that certain cell types are lineally related. And what this also allows us to do is to profile many, many progenitors at the same time in the same culture, each with a different barcode. And it also eliminates the need for clear spatial segregation between our clones. And also to reiterate, this complementary strategy tells us what the cells, what the fate of the cells are, so what they will become versus what they can become. So based on our experiments, we've analyzed approximately 50 progenitors, we've labeled 50 progenitors, and we've analyzed 176 cells that arose from those progenitors. We can detect 29 single-cell clones, or in other words, 29 unique barcodes. And after some filtering, we have 8 clones that span multiple cell types. So shown here again is the lineage prediction, color-coded by time. So the cells that we're labeling, just to be clear, is represented in green, and the cells that we're analyzing are represented in purple. So of those 8 clones, we see that seven indicate that these two cell types are related, and 1 clone indicates that these two are related. And most importantly, this shows that our lineage predictions are consistent with what we see empirically. And we also do not see any lineage relationships that are not predicted. So I'll now hand it back over to Boaz for some final words.
So to summarize the presentation, we've established reproducible methods to produce excitatory and inhibitory neurons as well as progenitors. We've been able to engineer stem cell lines for isolation of defined populations of cells. We've generated single-cell transcriptomic data to profile these cells at high resolution, and it's something that hasn't so far been shown in the field, and I think is very important for the field to do to understand what these cell types are. We've compared these, which is also something that's not commonly done, to actually developmentally relevant tissue from atlases and primary tissue, and demonstrated that we see both the rostral and the caudal cell types. We've inferred a computational lineage tree from this expression data and show that it captures this rostral and caudal split. And we're beginning to validate this lineage tree with both fate and potential mapping strategies. So I would like to end by thanking the in vitro human cell types group. Most of them are pictured here. They've been a really wonderful group of people to work with. As the stem cell portion of this group is coming to an end, and some of these people are going to be moving on. I wish everybody the very best. I'd like to thank Sharad Ramanathan as well as John Phillips for leadership. And lastly and importantly, we'd like to thank the Allen Institute for Brain Science and Paul Giallen for his vision and support. Thank you. Other questions?
Just, if you guys wouldn't mind, if you're asking questions, just to stand up.
What do you see as the medical applications? It seems like there might be some not that far away. Which ones would be the first ones to shoot for?
I think there are a lot of potential medical applications for this kind of a system. I think the medical applications are based on how accurately you're representing the thing that you're interested in studying. So there's certainly people who are interested in taking some of these cells and trying to engraft them in a regenerative therapy. You could use them for testing drugs on them if a process that you're particularly interested in is present in these cells. I think there's numerous potential applications. Mm-hmm. Oh.
Hi, Boaz. Is this on? So given that there are a lot of groups out there doing similar work in the mouse, trying to use similar models to piece these kind of tiny transitions in lineage together, what do you think you've done here to really open up understanding as to what makes us or makes the primate unique in terms of understanding lineage? I'd be really interested to know if you have an opinion on that.
Yeah. I think where we're actually providing the most insight is I think we've gone farther in depth into these processes than any published reports that I've seen so far, and so I think we have as firm an understanding of exactly what we're making. What's interesting too, and we didn't present here, and we have a paper that was recently published in Nature Methods where we showed the identification of outer radial glial subtypes. And these are progenitors that we're actually beginning to identify in these cultures, especially at the later stages. So although I don't have any particular insights that I can put my finger on, I think this is going to be a very good model, potentially for understanding how this protracted period of neurogenesis occurs and what these specific types of progenitors are actually giving rise to. And I think that's a big question in the field. I think Evan?
Yeah, interesting. I don't know if you can hear me. Oh, you can. Interesting
can
work. I was curious, it sounds like you had this one primary data set to compare to, and I was wondering, obviously there's a dearth of available data sets to compare to, but I wondered how well you feel you can date the 54-day-old progenitor cell types.
So that was one of the major objectives. I think where we date these cell types to is somewhere between late first trimester and early second trimester at day 54. We've carried out the protocol through 120 days, and we start getting longer and older cell types, but I think it's still very premature. I think this is a major hurdle for the field, is trying to figure out how to mature the cell types properly, and if there's some special sauce that people are missing.
Hi. Have you thought of doing the single-cell RNA-seq profiling on the human gestational tissue to see how accurately you recapitulate the in vivo setting?
And yeah, and we do that. We have a small data set currently from mid-gestation. We see a reasonable, I think, from progenitors to neurons, they're very easy to distinguish, but I don't know, Zijan, do you want to answer this? Or
Yeah,
I think she's...
we see a progression of progenitors to intermediate progenitors and neurons in the fetal tissue single cell data, but there seems to be a lack of further diversity within the neuronal cell types at that early stage we're capturing, which is post-conception week, 100 days or so.
Yeah.
Yeah, so we basically captured similar phases in our in vitro cell tests. Of course, we're still lacking some like mature neuron markers, but it seems to follow the similar trajectory, but kind of a little bit trapped in the early stage of development. Yeah.
Great. Thanks.